Have you ever wondered how our body gets proteins to make energy? Just like a power plant that produces electricity for our cities, our bodies rely on proteins for necessary function. Proteins carry out a variety of jobs such as breaking down your food, producing antibodies to fight infections. Proteins are made up of several amino acids assembled together. These amino acids are assembled using a special molecule called a ribosome. However, there's a special type of ribosome called a mitochondrial ribosome, aka mitoribosomes. These are located within the mitochondria, the powerhouse of the cell, and are in charge of making proteins only for the mitochondria. The mitoribosome has two parts, the small subunit and the large subunit. Both of these subunits are made from proteins and ribosomal RNA. The small subunit binds to the mRNA, which contains instructions to make proteins, while the large subunit aids in the formation of peptide bonds between amino acids. Let's take a closer look on how these two subunits work in humans. First, a fully processed mRNA is transported into the mitochondria. Within the mitochondria, the small subunit binds to a tRNA on the first ribosome to form a complex. This complex then binds to the 5' cap of the mRNA and scans it for the start codon. At the start codon, the large subunit binds to the complex. There are three sites within the large subunit, the E site, the P site, and the A site. The E site is where the tRNA exits the mitoribosome. The P site is where the peptide bond is made, and the A site is where the next tRNA attaches. During elongation, the first tRNA moves to the P site, and the subsequent tRNA with the complementary RNA attaches to the A site. Then, a peptide is formed between the two amino acids. Afterwards, the tRNA for the previous amino acid shifts to the E site and leaves the complex. The second tRNA, which now has a chain of amino acids, moves to the P site and the next tRNA moves to the A site. The process repeats itself until the mitoribosome reaches the stop codon, which signals the end of the translation process. In termination, a release factor binds to the A site instead of a tRNA and cleaves the bond between the growing peptide chain and the tRNA, freeing the newly birthed protein. This process is done by thousands of mitoribosomes in each of the cells across the entire body. These mitoribosomes collectively allow cells to produce enough energy to continue their daily functions.